My name is Dan Barouch. I'm at Beth Israel Deaconess Medical Center in Boston, and I'm one of the co-PIs of the I4C Collaboratory, along with Nelson Michael and John Mellors. And we are delighted to participate in this year's pre-CROI community cure workshop. And I'm very happy that you were able to still hold this virtually despite the current situation. I will try to give an overview of our I4C Collaboratory. I'm planning to leave substantial time for questions and discussion. And I think that in I4C, as well as I'm sure the other collaboratories as well, then we really integrate the community advisory board with the science that is being done. So the title of our collaboratory is Combined Immunologic Approaches to Cure HIV, or I4C, Immunotherapy for Cure for short. Okay, do you see my second slide that says strategies for HIV eradication on the top?
Yes, we do.
Okay, very good. So I think we're in business. So there are actually many strategies for HIV eradication that are being pursued by the field. It's a large, complex field with many, many groups trying to solve this important problem. Here is a partial list of strategies that are being pursued. The early institution of antiretroviral therapy is being pursued as a potential strategy for cure. The development of reservoir activators, also known as latency activators, is a key part of the field. Immunomodulators is another key part of the field. Gene therapy is a very exciting strategy, and I believe after me, James Riley will be speaking on some of the really cutting-edge work on gene therapy for HIV cure. Bone marrow transplantation is the only strategy that has actually worked for HIV cure. We have at least two, possibly additional, but at least two examples where there is very convincing evidence of either long-term remissions and possibly true cures of HIV infection through bone marrow transplantation in individuals who needed a bone marrow transplant for other reasons. Unfortunately, bone marrow transplantation in and of itself is not scalable and cannot be applied to the large numbers of people in general with HIV, but nevertheless provides a proof of concept that we think that an HIV cure is probably possible. Our own interests in I4C are in the immunologic approaches, namely broadly neutralizing antibodies and therapeutic vaccines that are shown in red. So our collaboratory is one of 5 collaboratories in the field, as well as many other groups working on HIV cure. We're now in year 4 of a 5-year program, and we're hoping that this can continue even beyond the 5 years. The goal of I4C is to evaluate active and passive immunologic strategies to target the viral reservoir in both the animal model, SHIV-infected rhesus monkeys, as well as, most importantly, in humans, in HIV-infected humans. I have the privilege of co-leading this collaboratory together with John Mellors and Nelson Michael, as well as a large number of senior investigators really throughout the world. Our collaboratory is very international with collaborators on multiple continents, including the Americas, South Africa, and Thailand. The overall hypothesis for I4C is that a combined immunologic approach that optimizes antiviral, humoral, and cellular immune responses, together with latency reversal, will reduce or control the viral reservoir in ART-suppressed SHIV-infected rhesus monkeys and in ART-suppressed HIV-infected humans. So we have many different strategies, But the overarching strategy really is to fall into two parts. First, we think that an immunologic approach might be able to rapidly eliminate the majority of virally infected cells. And this potentially could be achieved by the combination of broadly neutralizing antibodies with latency reversal. The concept here is that broadly neutralizing antibodies are incredibly powerful and can seek out cells expressing HIV envelope. However, that alone might not be enough because a latent cell that has integrated provirus and has no expression because of its latency would not be susceptible to an antibody. So if you combine a broadly neutralizing antibody with an immune activator that might either reverse latency or just cause generalized immune activation, you might be able to stimulate the reservoir cells to the point where they might express sufficient HIV envelope to make them susceptible to the antibodies. So that's why one of our strategies is to combine broadly neutralizing antibodies together with an immune stimulant that we hope will reverse latency. But we are full aware that that probably is not going to be sufficient because there is probably no chance that we would be able to eliminate every single provirus containing cell in the body. So we think a long-term component is needed as well. So for residual virally infected cells, we think there needs to be an element of long-term immune control. And for that, we believe that therapeutic vaccines might have the capacity to augment immune surveillance and therefore give a degree of long-term immune control as well. So our approach has been to test both individual and combinatorial approaches of passive immunization with broadly neutralizing antibodies, or DNAbs, as well as active immunization with vaccines. And for this, we are engaged in interactive and iterative non-human primate studies and clinical trials with additional focus on basic reservoir biology. Just want to make sure, are people still with me? The slide should read I4C Community Advisory Board.
Yep.
Yeah, carry on.
Great. So in I4C, we have a very active community advisory board that involves all 4 of our clinical sites, which is Boston, Pittsburg, South Africa, Durban, South Africa, and Bangkok, Thailand. Our CAB members are highly invested and highly committed. We've had a very stable, very energetic CAB for over 3 years now. Even prior to joining the I4C CAB, these individuals have experience working either as research participants or on other community boards, and all of them have worked in their communities for many years. So we are really privileged to work with this group of individuals. Our CAB representatives attend multiple scientific conferences, as well as collaboratory meetings, such as the NIH hivcure meeting, the HIV persistence meeting, etc., and are present on virtually all of our I4C investigator calls. And there's also numerous communications within the CAB that occur between the monthly full I4C calls. This is just a partial list of some of the I4C CAB activities. And this is a slide made not by me, but by the CAB in their presentation. So thank you for that. Our CAB has engaged in really a 2-pronged approach to community engagement, both dealing with the challenges and opportunities of having such a geographically separated, disparate CAB involving 4 sites across 3 continents. So they come together and they use extensive use of social media technology to reach people on a larger scale, as well as to help bridge some of the geographical distances between them and their communities. The websites are updated regularly. Webinars are recorded and uploaded to the website, and there are multiple different activities. Here are some from the IAS meeting in 2018, as well as a number of meetings since then. The second pronged approach is community engagement at the site level, and this is where each site representatives to the CAB host individualized events. Bangkok site has hosted HIV vaccine events. Pittsburgh site has participates routinely in Pride and provides education on HIV cure related research, and similar activities at the Boston and Durban sites. Much of the work of the CAB has focused on ATI guidelines, really together with the CAB members of all of the Delaney collaboratories, in terms of review of ATI criteria from the Consensus ATI workshop that was held about a year and a half ago. Our CAB provides input on our clinical protocols, and there was a very good discussion at the Miami Persistence meeting several months ago, where the I4C CAB, together with the CABs of all of the Delaney collaboratories, got together with the PIs for important discussions on the role of CABs in the clinical trials, the debates concerning ART treatment interruptions, as well as moving forward how CAB members could potentially travel with protocols from the more research-based collaboratories to protocols that might eventually be conducted in the context of clinical trial networks.
Dr. Barouch, I was wondering if you could explain what an ATI is just very quickly.
Oh, yes, absolutely. So sorry about that. So, sorry, that's an acronym. That's antiretroviral therapy interruption. So in the context of cure studies, actually this goes for both non-human primate studies as well as human clinical trials. A typical study might involve enrollment of either HIV-infected humans on ART, on antiretroviral therapy, or SHIV-infected rhesus monkeys on antiretroviral therapy. Interventions are then implemented, be they gene therapy or antibodies or otherwise. And then after the intervention period, then treatment is discontinued. So that's very important because although the virologic measures are improving in terms of how we can measure the viral reservoir, there is still no perfect measure in the laboratory of the viral reservoir. And to know whether interventions are successful or not, then one of the clearest measures is to stop antiretroviral therapy and to see whether virus rebounds. So stopping antiretroviral therapy to examine whether viral rebounds in a controlled, safe research setting where participants are monitored extensively is called an antiretroviral treatment interruption, or ATI. Does that make sense?
Yes, great. Thank you very much for that.
Yeah, sorry for the acronym.
No worries.
Okay, so now I'm on a slide. The title is Report of Consensus ATI Workshop. So I'm glad you asked. Thank you for your question of ATI, because I continue to use it. So this was a meeting that involved a large number of people. You can see from the author list of this article, there was a consensus workshop on essentially how to do antiretroviral treatment interruptions in an ethical and safe way. Understanding that there is a low level of risk for the participants, but it generates critical scientific knowledge that cannot be generated otherwise for evaluation of HIV cure strategies. And the author list is just a partial list of the participants of this meeting, which was held at the Reagan Institute. I believe it was the fall of 2018. And then this report from the meeting was published in Lancet HIV in April of 2019. And as you can see, this involved investigators, clinicians, scientists, community advocates, CAB members, funders, stakeholders is really quite a broad representation of individuals, multiple different stakeholders, including many of the people on this call today. And so this was a consensus recommendations for how to safely and ethically and rigorously conduct analytical antiretroviral treatment interruptions in the context of HIV trials. And here's the reference. I'll leave it to everyone on this call who hasn't read this article and is interested in this to read this. It really represented a coming together of many different stakeholders in the field, and I think has really set guidelines for how ATI studies can, should, and are being conducted currently.
Dr. Barouch, we had someone ask if you could comment on the clinical downsides of the ATIs that we learned from 10 years ago, is the question that Natalia is asking.
Sure. So there have been ATI studies conducted in various forms really since the initiation of antiretroviral treatments. What has been learned is that it is generally unsafe to take people off medications and leave them off medications for prolonged periods of time. That can then lead to adverse clinical outcomes. And so what has been done is close monitoring of individuals following treatment interruption. And if individuals are monitored closely in a clinical trial setting with very clear ART restart criteria, then so far it looks like it's very safe. Now, the key thing is, what are the enrollment criteria for participants in the clinical trial, and what are the restart criteria? Those are two critical questions that investigators, together with the CAB members, need to grapple with when considering a new clinical trial. For example, in most studies, individuals who have a history of advanced AIDS, very low CD4 counts, opportunistic infections, generally are excluded from clinical trials for the fear that resurgent virus could lead to a rapid deterioration of clinical status. So there often are nadir CD4 requirements for enrollment in such clinical trials. The second, as well as overall health and history, basically we want to enroll individuals who are relatively healthy in these treatment discontinuation studies. The second element that's important is, what are the criteria to restart antiretroviral therapy? And this is where investigators have had quite a bit of difference of opinions in terms of in some studies, any level of virus detected, even very, very low amount of virus, leads to restarting of antiretroviral therapy. In other studies are a bit more permissive, where a certain amount of virus for a certain period of time is tolerated prior to initiation of antiretroviral therapy. And so that depends on the science of what's trying to be done, as well as overall guidelines of what is believed to be safe in the field. So I think safety is always, participant safety is always the most important consideration. And in addition to that, then the scientific question that's being asked is important. For example, if the goal of an intervention is to prevent rebound, then one can probably restart ART as the first signs of viral rebound. However, if one is looking at, for example, maybe a therapeutic vaccine where it's not really thought to prevent rebound, but it might control virus after rebound takes place, then it might be scientifically justified to have individuals be watched carefully with rebounding virus for a limited period of time prior to restarting antiretroviral therapy. So it's really a combination of safety first, but also taking into consideration the scientific goals of the study.
Great, thank you.
Great. So moving on, the next slide should say I4C Science on the top.
It does.
Yes, sorry, another question?
No, 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 it does. I was just saying, yes, we see it.
Very good. Yeah, thank you. So in I4C, we really are engaged in multiple different areas of HIV cure research, including basic research on the viral reservoir, how to measure it, how to characterize it, and how it behaves in the setting of rebound. We are also heavily engaged in non-human primate studies, animal studies, preclinical studies that help us define candidate cure strategies, explore hypotheses, and more appropriately focus the clinical trials into clinical trials that we think or we hope will have more impact on the field. And then, of course, we are heavily invested in early phase clinical trials to explore cure strategies in the most important model, which is human. We also have iterative integration of the basic, the preclinical, and the clinical science because we think that all three are important. They're basically the three pillars of cure research, and working together, basic research in the laboratory, preclinical studies, and clinical trials all need to be integrated to generate knowledge towards the development of an HIV cure. So I'm just going to show one slide of the basic research work. I4C is working in many different areas of basic research, but I thought I would highlight this one slide from Bob Silicano, who's part of I4C, as well as Bob contributes to other collaboratories as well. But Dr. Silicano at Johns Hopkins has created new assays to measure the replication-competent viral reservoir. He calls these IPDA assays, or integrated provirus development assays, integrated provirus DNA assays. And the purpose of these assays is to measure not just the integrated provirus, but the replication-competent intact provirus. Sorry, I misspoke. It's intact provirus DNA assays. And the reason why this is important is because most of the provirus in cells is defective. So traditional measures of viral DNA pick up a huge amount of junk. And so now there are PCR assays that pick up, basically can exclude most 95 or higher amounts of the defective provirus and can be a pretty good approximation for the replication competent intact provirus, which is what really matters in the end. So Dr. Silicano has developed these assays, which have really been an important contribution to the field and is a new way of measuring the viral reservoir, and therefore is important in measuring the efficacy of potential interventions. This assay has also been adapted to measure intact provirus from non-human primates. I'll spend a few minutes talking about some of the preclinical studies in non-human primates that the I4C has been doing. And one of the applications of the non-human primate model has really been to test therapeutic vaccines and broadly neutralizing antibodies with the idea that preclinical evaluation of these interventions could help us focus our clinical trial strategies. So a question that we have recently addressed in non-human primates is the central hypothesis of I4C, which is, can combined active and passive immunization together with an innate immune stimulant target the viral reservoir in ART-suppressed, SHIV-infected rhesus monkeys, and then either control or delay viral rebound following ART discontinuation. So on this slide is a typical design of an NHP study for HIV cure. So typically, to test an intervention for HIV cure in monkeys, we would infect the rhesus monkeys with a monkey version of HIV. This is called SIV for simian immunodeficiency virus, or SHIV or simian human immunodeficiency virus. After infection of the monkeys, then we would initiate antiretroviral therapy, or ART, to suppress viral replication. And after a certain period of time, typically a long period of time, we would begin an intervention, such as a therapeutic vaccine, such as a broadly neutralizing antibody, such as a latency reversing agent, as well as all the other interventions on the list I showed you at the beginning of this talk. Then there would be important measurements of the viral reservoir using the various assays that I just discussed, such as viral DNA assays, perhaps the more advanced IPDA assays, as well as other assays. And then after another period of time, then typically ART will be discontinued. And off ART, the key outcome is to measure viral rebound following ART discontinuation. For example, we could ask, do animals rebound at all? If so, what is the timing to rebound? And what is the level of virologic control? Eventually, we hope to be able to do studies that don't require ART discontinuations, perhaps if there is a perfect measure of the viral reservoir. We currently don't have such a measure. And so we feel that discontinuing drugs and assessing explicitly for whether virus rebounds or not is really still the most important outcome. Hold on one sec. I clicked on something and I've lost my own screen now. Hold on a sec.
Okay. Let's see, you want to go to your slides. So move that window down. Oh, wait. Public computer option. I don't know what this is. Secure remote access.
Are you still seeing my slide, or?
No, we're not. We're seeing whatever's on your screen. So what you see on your computer is what we're seeing. So we're seeing that
Okay.
this BIDMC.
Okay. No, I got a pop-up box and I clicked OK and then, okay. Do you see my screen again?
Yes, we do.
Thank you.
Okay, great. So whatever is my screen is what you're seeing.
Yes.
That's correct.
Okay, great. All right. This is going much better than I had anticipated.
Good.
Thank you for your patience.
You're doing great.
All right. So I'll take you through two studies and just the high points of data. I'll take you through two studies that have been published and then one study that is not yet published, which is actually my presentation at CROI, which was supposed to be on Tuesday and which I'm trying to figure out how to do the audio presentation for. So the first study that was published in 2016 was an analysis of a therapeutic vaccine. The therapeutic vaccine involved Ad26 and MVA vectors. And we also looked at an innate immune stimulant, which was our TLR7 agonist. It has subsequently been called Vesatolimod, or VES. And the TLR7 agonist was provided to us by Gilead. So in this study, we infected monkeys with SIV, we treated them with antiretroviral therapy, and then we administered either the TLR7 agonist, the vaccine, or both. And then we stopped drugs. We stopped antiretroviral therapy. And what you see here are viral loads in these four groups of animals after ART discontinuation. As you see here, all the animals and all groups rebounded. So none of these animals were cured. The sham control group, these animals rebounded very quickly and very robustly. In the group that received the TLR7 agonist, there was a very similar profile of viral rebound. In the group that received the vaccine, the Ad26 MVA vaccine, there was a very similar profile of rebound as well, but a few of these animals appeared to control virus to lower levels. And then the animals that received both, shown at the bottom right, these animals had a slight delay of viral rebound, but more importantly, there was a more convincing reduction of viral loads there was a reduction of set point viral loads in almost all animals. And interestingly, 3 of 9 animals shown with a red arrow showed virologic control to undetectable levels during this time period. Now, these animals rebounded, so these animals are not cured. So this is a phenomenon of post-rebound virologic control that was correlated to animals that had some of the highest T-cell responses induced by the vaccine. Now, these observations, I think, do have some implications for the design of clinical trials. For example, in a therapeutic vaccine study in humans, if we are obligated to start antiretroviral drugs immediately upon evidence of viral load rebounding, then there would be no way we would ever see this phenomenon of post-rebound virologic control. Of course, that has to be balanced against the safety aspect of letting people continue with replicating virus for a period of time. But I think these data, nevertheless, are thought-provoking. So let's turn now to antibodies. There are many different antibodies against HIV. The broadly neutralizing antibodies, or DNAbs, are typically clustered into the different epitopes that they react with on the HIV envelope surface. They have very strange acronyms and numbers and letters. I will just concentrate on one antibody today, which is against the V3 loop, which is the purple blob on the corner. And the antibody is called PGT121, which is a potent glycan-dependent antibody that binds the third variable loop, V3, on the HIV envelope surface.
Dr. Barouch, could you tell us, when you were looking at that illustration, what was that we were looking at?
Oh, sorry.
Those blobs.
So, yeah, so the light cyan blob is the HIV envelope protein. It's on a pancake. The pancake on the bottom is sort of the viral envelope surface.
Okay,
and
got
then
it.
The colored blob in darker blue, yellow, purple, red, and green are structures of antibodies that are bound to the HIV envelope in cyan.
Got it. So this is like a minuscule, miniature replica of just one of those little stubbies that comes out of the HIV virus.
Exactly. So here we're showing how 5 different antibodies bind, the positions of their binding on the HIV envelope protein.
Got it. Thank you.
So here are data from another study that we published. It was our first study of a broadly neutralizing antibody in the monkey model. It was published in 2018. And this is a study of the antibody, the broadly neutralizing antibody called PGT121, the one that was binding to sort of the corner, the purple blob in the corner on the previous slide, together with the same TLR7 agonist, Vesatolimod, that was in the first study. So here, the studies were conducted in a similar way. Animals were infected with a virus called SHIV. Then the animals were placed on antiretroviral therapy. And then animals received either the TLR7 agonist, the antibody PGT121, or both. We waited a period of time to let the antibody titers wash out, and then we stopped antiretroviral therapy and looked for viral rebound. In the sham control group, all the animals rebounded. In the group that received the TLR7 agonist, 10 out of 11 animals rebounded to similar levels. In the animals that received the antibody alone, PGT121, 9 out of 11 animals rebounded, but some appeared to rebound later into lower levels. And most importantly, in the animals that received PGT121 plus the TLR7 agonist, only 6 of 11 animals rebounded. And these generally occurred later into lower levels. So these results are shown most clearly by this Kaplan-Meier curve, in which we show the time to rebound. The percent of the animals rebounding is on the y-axis, and the days following ART discontinuation is on the x-axis. You can see the sham controls in black, all 100% rebound quickly. In the animals that receive the TLR7 agonist in green, were very similar to the controls. The animals that received the antibody alone in red showed a slight change of this curve, but clearly the most important effect was seen in the curve shown in blue, which is the animals that received the antibody together with the TLR7 agonist in terms of a delay and in some cases a prevention of viral rebound. So some new data that we're presenting at this year's CROI conference, which I'm happy to give you this preview and basically the high points of the data, is our study in monkeys looking at combining active and passive immunization. Active immunization is the vaccine and passive immunization is the antibody. So this study clearly builds on the prior studies that I just mentioned of the vaccine alone and the antibody alone. In this study, we used 51 rhesus monkeys that were infected with SHIV. We then initiated antiretroviral therapy on day 9, tenofovir, FTC, and dolutegravir. The vaccines were Ad26' MVA boost vaccines. The antibody was 5 infusions of PGT121, and the TLR7 agonist, as in the prior study, was the Gilead molecule Vesatolimod or VES. Group 1 received the vaccines and the antibody with VES. Group 2 received the vaccines with VES. Group 3 received the antibody with VES, and Group 4 was the sham controls. So here are the viral rebound data. Similar to the previous study, animals were infected, placed on ART, given the interventions, and then we stopped drugs. So this is what happened. In the sham control group, all the animals rebounded as before, which is not a surprise. If we look on the lower left, the Ad26 MVA vaccine plus VES, all the animals rebounded, as we saw previously, but set point viral loads appear to be lower. And here we see this phenomenon again, where 3 out of 12 animals had this phenomenon of post-rebound virologic control. If we look at the upper right, the antibody PGT121 with VES, only 8 out of 12 animals rebounded, and 4 have remained negative through day 84 following treatment discontinuation. Again, similar to what we've seen before. And the new part of this study is when we combine vaccines with antibodies, then we appear to have an additive effect where only 6 out of 10 animals rebounded. And of the animals that rebounded, viral loads tended to be lower. And we saw an additional 2 animals that did rebound that subsequently controlled. So actually 6 out of 10 animals had undetectable viral loads by day 84 following treatment discontinuation. So again, this is shown probably most clearly by the Kaplan-Meier curve shown on the left, in which the blue and the red lines, reflecting the 2 groups that received the antibody, clearly had a delay of rebound compared with the sham group and the vaccine-only group. And these were significant by many different statistical tests that I don't need to go into today. So the conclusions from this new study is that active immunization plus VES resulted in post-rebound virologic control, and passive immunization, which means antibody infusion, resulted in delayed viral rebound in a subset of the animals. Combined active and passive immunization with the TLR7 agonists resulted in both delayed viral rebound and post-rebound virologic control, suggesting that these 2 immunologic interventions can be used with additive effects. And we think taken together, these data suggest the mechanistic basis of how humoral and cellular targeting strategies might be able to have an effect in the context of so-called shock and kill strategies. So I'll turn now to clinical studies. Our teams through I4C and the expanded groups that we come from and represent have engaged in a number of different phase 1 clinical trials. So I have used some slides from my colleagues, first from the MHRP group, led by Nelson Michael, Jintanat, Denise Xu, Sandy Vasson, etc. They have tested a number of different interventions in their acute cohort in Thailand. A first study, RV411, looked at ART alone, and then stopping ART following treatment of acute fever-1 infection. All individuals rebounded. So they moved on to looking at interventions, including the potential LRA called vorinostat, as well as looking at a broadly neutralizing antibody here given after treatment was discontinued called VRC01, and most recently RV405, which is a therapeutic vaccine, the Ad26 MVA vaccine given prior to treatment interruptions. In general, the effects following treatment interruptions have been modest, shown in an example on the bottom right slide. There have been effects with a very slight delay of viral rebound, but nothing that approached a complete prevention of rebound. So I think the thought is that there needs to be combinations. And actually, if we think about the monkey studies, antibody alone and vaccine alone had a minimal effect in their own right. So if we want to trust the preclinical animal data, then we would suspect that combinations of immunologic interventions with immune stimulating or latency reactivating strategies are going to be needed. Another clinical study that's being supported through I4C is a study that we hope to start soon, being led by John Mellors, Sharon Ridler at University of Pittsburgh, together with the MHRP group, Denise Xu and colleagues in Thailand. And this will be a therapeutic vaccine study with a dendritic cell vaccine. And we would like to know how immunogenic this vaccine is, given that in I4C, we're really looking at several different vaccine strategies, the vector-based vaccine strategies and the dendritic cell-based vaccine strategies. At our own institution at BIDMC, we have concentrated on a series of broadly neutralizing antibody studies. We did the first in human study of the antibody PGT121, and we showed that it's safe, it has good pharmacokinetics, and it has good antiviral effects in viremic HIV-infected individuals. Probably the more interesting study in terms of cure is a study that's just started now, that's ongoing, and it's recruiting right now, which involves the use of a triple antibody cocktail, because no single antibody will cover all the viruses that are out there. And the goal of this study is we're taking HIV-infected individuals on ART with suppressed virus, we're giving them the triple antibody cocktail, and then we're stopping therapy, and we're continuing to administer the antibody at monthly intervals. And we've so far enrolled 2 individuals in this study, and they are doing very well, and we hope to enroll 12 individuals in the treatment interruption group in group 2. And so we hope that in the coming year, we'll have data from this study that we will be able to share with you. In addition, I4C, we cannot do everything ourselves, of course, and so investigators from I4C are leading several key proof of concept studies, mostly involving broadly neutralizing antibodies, together with the ACTG clinical trial network. These studies involve collaborations with the VRC, Division of AIDS, and industry. These trials are a natural extension of the science done in I4C, the in vitro and NHP studies, as well as the early phase clinical trials. These studies with the ACTG network leverages expertise and resources of all stakeholders and exemplifies the collaboration and rapid product evaluation. There are at least 3 trials that are being led by I4C investigators within the context of ACTG. 5387, which is a dual BNAb combination with a TLR9 agonist. 5388, which is a dual BNAbs with ART and acute infection. And 5389, which is dual BNAbs and acute or chronic infection. And with the next generation of the Delaney collaboratories, then I believe that my understanding is that this is intended to be the model where some of the science is developed within the collaboratories, and then clinical trials are conducted by outside funding sources and groups, either the ACTG network, individual sites, UL1 grants, etc. And I should mention there was a very good discussion at the CAB meeting in Miami, at the Miami Persistence meeting, about how these studies are best conducted, and offered a strategy for longitudinal CAB involvement in the context of collaboratory ACTG collaborative studies, where the CAB members can participate in the Delaney collaboratories and then travel with the protocol to the clinical trial networks and participate in those, in the conduct of those studies in the networks. And I personally think that's a very good idea and hope that can be implemented. So I'll summarize briefly, and I think we still have 5 minutes for questions. Within I4C, we have shown that both therapeutic vaccines, as well as broadly neutralizing antibodies with an innate immune stimulant, can impact viral rebound following ART discontinuation in non-human primates. Our new data shows that combination studies involving vaccines plus antibodies and immunomodulators can show additive effects. Phase 1 clinical trials with multiple different therapeutic vaccines and DNAbs are in progress or planned. This is true in I4C as well as in the field in general. And for all of our clinical studies, our CAB has a very critical role to play. And we're very appreciative of them. We also in I4C conducting basic research on improving our understanding of the viral reservoir and our ability to quantitate the reservoir. So I'll stop there, and happy to take any questions for the last 5 minutes.
Great. Thank you, Dr. Barouch. We do have a question that David wanted to ask. It's, do you believe that requiring laboratory criteria demonstrating at least a minimal amount of new immunity or HIV protection, et cetera, should be required to initiate an ATI?
It's a very good question. I think it has to be context specific. So I think it has to be individualized for the particular trial. For example, if we're testing a vaccine, and if theoretically and preclinically we believe that the mechanism of action is due to the vaccine inducing potent T-cell responses, and if the goal of the vaccine is to induce potent T-cell responses, then I do think that there should be some measure of T-cell responses in a clinical trial prior to initiation of an ATI. Now, obviously, the levels, the time points is all debatable, but theoretically, if a vaccine that is believed to work through T-cell responses generates no T-cell response whatsoever in humans, then it's very difficult to justify the additional effort, cost, risk of doing an ATI if the product did not achieve its intended purpose. Similarly, if one is testing an immunomodulator or an immune activator, one would want to see some scientific measures of immunomodulation or immune activation, essentially to show the product had the intended effect in the individuals. And probably if it has no effect whatsoever that can be detected, then the risk benefit and the scientific justification for proceeding with the ATI is reduced. However, there are some interventions that may not have a clear laboratory measure. And for those, there may not be a clear measure that we are confident about as to whether the product had the intended effect or not. And for those, then I think that there in some cases might be a scientific justification to proceed with an ATI regardless. For example, in the early stages of early ART therapy alone, there's really nothing to measure, and there is no knowledge as to whether those individuals were cured or not. The only way of doing it is to stop therapy and see if people rebound. So I think that the answer to your question, it's an excellent question, and I think it has to be context specific. It has to be related to the science of each individual trial.
Thanks, Dan.
We have a question.
Any other
Jessica.
Any other questions?
Yes. One's coming from the line of Jessica Saltzwedel. Jess, are you there?
Yes. I'm not hearing any questions.
Yeah, maybe if Jessica can't unmute, maybe I can sort of try and read it from the chat. I think it was a question about whether any possible variation in how different clades of HIV behave, like if some might be, replicate more strongly than others, if those would affect sort of immune-based strategies, particularly if you're working internationally.
I would say that the clades and viral variation more generally has a very important effect on the immune-based strategies. And it's not solely replication, but it's also viral diversity. For example, there is so much diversity in HIV, we have to make sure that the vaccines or the antibodies we're using are relevant for the circulating viruses in a particular region. Fortunately, in our therapeutic vaccines, we're using so-called mosaic inserts that do raise a greater degree of cross-reactive T-cell and antibody responses against multiple clades. And for the broadly neutralizing antibodies, it's very important to use antibodies that are relevant. For example, the V3 antibody PGT121 has virtually no coverage of clade AE. So it would not make sense to do a PGT121 study in Thailand. That would just not make scientific sense. And why for antibodies, most people are moving towards antibody cocktails. And with a triple antibody cocktail, then one could get 99% coverage of global viruses. But with a single antibody and even a double antibody cocktail, then there are holes in coverage. So the question of virus diversity in a clade as well as in a particular population or even a particular individual are really important. And one needs to make sure that the interventions have relevance for the actual sequences of the virus in the population that's being studied.
Thank you.
Thank you.
Like Jessica's microphone isn't working. Jess, if you had any other follow-up points to Dan's response, please send them in the chat box. Were there any other questions? Michael, you mentioned an additional question as well.
Yeah, Mark Wagner had a question about drug metabolism and ATIs.
Yes, what is the question?
I think I don't know if Mark can unmute and ask.
Correct. If not, there's a question from Simon. Simon is asking, what is likely the coverage of VRC01 in the AMP study in Southern Africa?
That's a very good question. The short answer is we know that in the test tube, there's about 70 or 80% coverage. However, we don't really know how those lab tests relate to humans, because in humans, there's many other complicating features, such as viral diversity, minor resistant species, recombinant species. So there are many factors in humans that might be more complicated than the virus neutralization assays that are being tested. So we are going to get the answers to the AMP study this year. So in a few months, I believe that the results are going to be available in late summer and are going to be presented at the R4P meeting this fall. And those data are going to be very important because we will see the degree to which VRC01 protects humans against HIV infection, both in clade C in Africa as well as clade B in the Americas. And that's going to be a very good gauge. And we will then learn whether the in vitro neutralization assays are predictive of protection in humans, and if so, whether they're even quantitatively predictive or not. There is some concern that the results of the AMP study, it might show that protection in humans is more difficult to achieve than simply coverage in a virus neutralization assay because of the complexities. But no one knows the data, and we will learn that. We will basically learn the answer to your question in a few months.
Thank you for that. It looks like Mark was able to type his question in the chat box. For some reason, folks are having challenges with microphones. We will address those logistical issues as the meeting unfolds. But here's Mark's question. It says, for our CAB presented recently to NIH about the role of drug metabolism effect on ATIs via the cytochrome P450.
I think maybe is that sort of related to whether people should stop drugs at different times before an ATI?
Yes, and I want to acknowledge and congratulate Mark and as part of the I4C CAB for bringing this important issue to everyone's attention. I'm not sure there's a question there, but we are certainly very supportive of that. And it was really an initiative of our CAB, Mark in particular, to acknowledge and emphasize the potential role of drug metabolism and how that could impact ATIs. And so within I4C, we don't have pharmacologists. But John Mellors has brought that issue to the ACTG, and I believe that they are very appreciative of the conversations. And Mark, I'm not sure if there's additional questions, except for the fact that we greatly appreciate your involvement and bringing this important issue to everyone's attention.
Thank you. And Mark says thank you in return. Any other questions?
Well, Dr. Barouch, I do have a question. So I love that your collaboratory spans 3 continents, and I'm sure that brings up lots of challenges, but I'm wondering whether it's from the community engagement side or the basic science side of your collaboratory. Are there any sort of unique ways that the differences have actually added to the pursuit of community engagement or science? Having to straddling from Bangkok to Durban just seems like there'd be lots of opportunities for learning.
Yeah, absolutely. And, perhaps some of the I4C CAB members can comment directly, but it has obviously presented logistic challenges, just finding a time of day for conference calls. If you have to include time zones in Bangkok, Durban, and the East Coast of the United States, obviously is a challenge. But, with today's social media and networking, then, I think they very effectively found ways of communicating and overcoming that, as well as to have discussions go to communities on 3 continents simultaneously. The in-person activities, of course, is challenging because obviously it's difficult for people to travel across the world for an in-person event. So while the discussions and social media engagement and online trainings can very effectively span 3 different continents, then the in-person trainings often are led by the CAB members at each site individually.
Great. Richard, how are we doing on time?
We're about 7 minutes over.
Okay. Well, go ahead.
If there was enough, it should be a very brief question and response. Can you say a bit about the compensation your CAB members receive? And then we can transition out of respect for time for our next presenter.
Sure. Very, very briefly, our CAB members are compensated. I don't recall the exact dollar amount, but certainly we greatly appreciate their time and effort, and all of their travel expenses are fully paid for all the conferences they attend as well.
Great, thank you.
Great, well, thank you very much. I'm glad that the audio and visual has worked. So I appreciate your ability to move this to a virtual meeting.